So what is the physical seat of thought? What is the source of our emotions, our decision-making, our passions, our pains, and everything else? Well, it's the brain, and it's said to be the most complex mechanism in the known universe. And you might expect, given all it is and given all it does, that it would look very pretty, full of shimmering lights and glass tubes and mysterious colors. But in fact, it looks really kind of gross. It looks like a 3-day-old meatloaf. It's gray when you take it out of the head, and inside the head it's bright red because of all the blood. In fact, it turns out, very surprisingly, that the source of our mental life, of our consciousness, is meat. In fact, you could eat it. People have eaten brains. I've had brain with cream sauce. Not human brain, mind you, but I've had brain with cream sauce. It's not bad. But it makes the puzzle all the more harder. How can this fleshy thing give rise to mental life? And that's the question I want to sort of explore in this lecture and the rest of the lectures. And I want to do so by starting with the smallest relevant parts, the different parts of neurons, then explore how the neurons are connected together, how they're wired up, how they form different subparts of the brain, like the hypothalamus and the frontal lobe, and finally talking about the brain in a larger perspective, looking at the two halves of the brain, the left half and the right half, and how they interact. Now, there's a lot of stuff in the brain, a lot of chemical stuff, a lot of different parts, but where the action is, the part that does the thinking, the part that is the focus of most of our research, is the neurons. It's not an accident they call the study of the biological basis of thought neuroscience, because it all comes from the neurons. So you can see here pictures of neurons interacting together. And here's a diagram that depicts a typical neuron. So what you see is the dendrites, and dendrites receive signals from other neurons, either excitatory, like pluses, or inhibitory, minuses. And then they get to the cell body, which sums up these pluses and minuses. And when you reach a certain threshold, a certain amount of pluses, there's neural firing. And the firing takes place through the axon. And the axon is much longer than the dendrites. In fact, for some motor neurons, it's very long indeed. There's axons running from your spinal cord all the way to your big toe. You could think of it, of the relative sizes of things, in terms of a basketball and a 14-mile garden hose. Surrounding the axon is what's called a myelin sheath. And a myelin sheath is, you could think of it as insulation, as fatty tissue, like insulation on a wire. So the information comes through the dendrites, it's summed up in the cell body, and it's transmitted through the axons. So what neurons do is they sum up and transmit information. And we know that there's a lot of them. By some estimates, it's 100 billion, or the estimates tend to be very different and very rough, but there's billions upon billions of neurons, and each connect to thousands, maybe tens of thousands of other neurons. And so the fact that you have something of this degree of complexity, this degree of structure which there's no way to replicate in any machine, the numbers are just too big, is why people might describe the brain as the most complicated machine in the universe. At least this is fitting. It's made of meat, maybe, which is kind of disappointing, but at least it shows this incredible internal structure. So neurons come in 3 flavors. There's sensory neurons, which take in information from the environment, from the external world. There's motor neurons, which go from the brain out to your motor control. So if you touch something hot and you feel a pain, that's sensory neurons. If you wrench your hand back or you reach for something, that's motor neurons. And finally, there's interneurons, which connect different neurons without making contact with the external world, either through sensation or through motor action. Now, the main thing to think about for neurons and neuron firing is that it's all or nothing. It's like firing a gun or sneezing. Neurons either fire or they don't. Now, you might think that's a little bit strange, particularly when you think about sensory neurons, because your experience seems to be a continuum. So you have sensory neurons in your eyes. And you could distinguish between a very dim light and a very bright light. You have sensory neurons in your fingers, and you could distinguish between gently touching something versus being stabbed on the tip of your finger with something. But still, the neurons are all or nothing. The way we get to this continuity of experience is that neurons can code for intensity in different ways. So one way is in terms of the number of neurons that fire. If X neurons corresponds to a mild experience, X times 10 neurons might correspond to an intense experience. Another factor is the impulse frequency of individual neurons. So an individual neuron might denote a mild sensation by doing fire, 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 while it might denote an intense situation with fire, 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 fire. So you have neurons, and the neurons talk to each other. They talk to each other because an axon of one neuron will communicate with the dendrites of another neuron. And a long time ago, people used to think that neurons were wired up together like a computer. But in fact, neurons don't actually touch one another. There's a gap between the axon terminal of one neuron and the dendrite of another one. A very tiny gap, typically of like 1/10,000th of a meter wide. And this gap is known as a synapse. And when one neuron fires, the axon releases neurotransmitters. These are chemicals that shoot out over that gap and affect dendrites on other neurons. And as I said before, the effect of these neurotransmitters could be excitatory, which is that they raise the energy, so they increase the likelihood of a neuron firing, or inhibitory, so that they bring down the likelihood of a neuron firing. And what's interesting is that different neurons shoot out different neurotransmitters. And so they have different effects on other neurons that they make contact with. And in fact, a lot of psychopharmacology, both attempts to cure various psychological or physical diseases by giving medicines, or recreational psychopharmacology designed to, increase pleasure of different forms, or sometimes help people work or help people focus, works by fiddling with the neurotransmitters. And these can be either antagonists that lower down the intensity of things by binding to the dendrites, making it hard to create more neurotransmitters, or they can increase the amount of neurotransmitters available in different ways. Agonists. So you're either sort of pumping up the volume or turning down the volume. So you can think about different drugs and their effects. There's curare. Curare is a drug used by South American Indians. It's an antagonist. It blocks motor neurons from affecting their muscle fibers. It keeps your motor neurons from working. And what that does is it paralyzes you. And in large enough doses, it kills you because motor neurons also keep your heart beating. So shut that down and you die. There's alcohol. Now, alcohol also has an inhibitory effect. You might think that's strange because when I drink alcohol, I get all excited and happy and goofy. And you have to keep this in mind here. The way alcohol works is it inhibits part of your brain that does the inhibition. So you have part of your brain that says, don't say that to the other person, keep your pants on, stop yelling. And alcohol basically inhibits that part of the brain, making you more exuberant. Then, over the course of things, in the course of drinking too much, it also inhibits other parts of the brain, so you could pass out and fall on the floor and in large enough doses die. So both curare and alcohol in different ways bring things down. Other drugs bring things up. So amphetamines, for instance, increase the amount of norepinephrine, which is another neurotransmitter, that's responsible for general arousal. And this is how drugs like speed or cocaine work. Other drugs like Prozac or L-Dopa influence neurotransmitters in ways that they increase, for instance, the supply of dopamine or serotonin, which can be relevant for issues like Parkinson's, which seems to be related to too little dopamine, and depression, which is related to too little serotonin. So these drugs work by influencing neurotransmitters, either by directly pumping in more neurotransmitters or increasing the supply in different ways, or stopping them from having effects by binding them or making it or sucking them up in different ways. But they work through their effects on neurotransmitters. So the more general idea is the way neurons lead to thinking is that they form clusters or networks. And these clusters and networks are computational devices that do interesting things like recognizing faces or walking upright or understanding sentences or doing math or experiencing great sadness or falling in love and so on. And we now know that that's possible because we create computing machines that work in certain ways. That if you wire up a computing machine in certain complicated ways, it can do mathematics, play chess, do flight simulator, and so on. And so you may be interested in a project of computational neuroscience, which tries to ask the question, how are neurons wired up to do interesting things, and uses our own success at computational theory as a model, and then sometimes takes the inference the other way around, which is you can see how people do it, and then use this knowledge of how people do it to create computational systems that can do it as well. So how is the brain wired up? Well, you might imagine that it's wired up like a portable computer, like a laptop, like the sort of computer you're looking at now. And to some regards it is, but there's a couple of reasons why it can't be. And both of them have to do with how well the brain works. So first, the brain is highly resistant to damage. If you get a knife to the brain, if you get damage to the brain, it won't typically shut down the whole system. The information and capacity are somehow distributed across neurons in such a way that makes them extremely resilient to damage. While in contrast, someone could open up the back of your laptop, pull out a chip, and the whole thing is ruined. The whole thing will stop working. But the brain is wired up in a certain way that makes it highly resilient. The second thing is the brain is wired up in such a way that it makes it work very fast. So computers can do millions of operations per second because they're purely electrical. But brain tissue is much slower and can't spend the time to do many steps. So to put it a different way, if your brain was wired up like a computer, it would be so slow as to be entirely unusable. It has to be wired up in a way that's more efficient, that allows for the slowness of brain tissues and neurotransmitters, and can still compute things at a human level, which is often blindingly fast. Because of this, there's been a huge interest in massively parallel systems and complicated neural networks, which are wired up as we believe the brain does. And as such, we're helping computers to do things based on their understanding of the brain that they could never do before. And the details of this is something we're going to talk about through the course. We're not actually going to end up explaining different capacities directly in terms of neurons, because we can't, and because we wanted to have higher level explanations. So when I talk about how people learn language or how they recognize faces, we're not going to talk much about neurons in particular, but we will talk about different brain areas and how they work. And then the assumption is, the bet is, that everything we talk about in more functional ways can ultimately reduce down to large networks of neural systems, and that in turn will ultimately reduce down to the specific behaviors of the specific neurons that we're looking at.